So I asked you the question, what stage does crossing over occur in? I'm going to ask you another one here in a minute. Let's see what you tell me. Prophase 1, right off the bat, we got it right. That's good. Let's see if everybody should have prophase 1. Not elephantmental, whatever it is. Okay. All right. Let me ask this. What stage does independent assortment occur at? At what stage does independent assortment occur at? I'll give you a hint. It's at one of the points at which they line up. Because that's when they get arranged, right? And 5, 4. You can even abbreviate like P1, M1, A1, T1, P2, M2, A2, T2. What would happen? I don't know. What'd I do? Oh, I got to go back. There. I don't. Now I broke it. No, you don't. I need to learn how to use my smartboard, though, a little bit better, right? It is metaphase 1. So, folks, keep those in your brains. Prophase 1 is where crossing over takes place. Then when you arrange them on right and left, that's where the law of independent assortment comes into play, because that will determine where they go. Okay? Does that happen again in the other metaphase as well? Think about it. Yeah, because you can flip the chromatids around the other way too. So we'll get into that more in chapter 12. That's the law of segregation. That's a different one. What? Oh, you're thinking like history stuff. No. So, see, when you say segregation in this class, I think of Mendelian genetics, not civil rights movements. All right. So the process of mitosis and meiosis are similar but meet different needs and have different results. In the last chapter, we talked about mitosing cells, like somatic cells dividing. In this chapter, we're talking about making gamete sex cells. You guys, I'm like not entertained by people not being on task. Get on task. Mitosis makes new cells that are used during growth, development, repair, and asexual reproduction, whereas meiosis is going to make cells that enable an organism to reproduce sexually, and it happens only in the reproductive structures. And if I ask you the reproductive structures, you would tell me those are ovaries and testicles, the male and female gonads. So mitosis produces 2 genetically identical diploid cells. We get that. We start off with 2N, we end with 2N. We start off with 46, we end with 46. That's mitosis. Meiosis, though, produces 4 genetically different haploid cells, and we want to be different. It creates more diversity. And if I ask you why they're different, you say, well, it's because crossing over happens in prophase 1 and because of the law of independent assortment that happens in metaphase 1. And if you compare meiosis to mitosis, they do appear similar when you start drawing the diagrams because they're both cells. We're dealing with chromosomes. We're dealing with dividing nucleus. However, remember, in meiosis, we're cutting the genetic information in half, and often we're doing it with more than one cell. Now, in prophase 1 of meiosis, every chromosome pairs with its homologue. And a pair of homologous chromosomes you learned yesterday is what is called a tetrad. Why tetrad? Because tetra means 4. Tetrahedral figures are 4-sided figures, right? So as the tetrads form, different homologs exchange parts of their chromosomes in the process called crossing over. Now the pairing of homologous chromosomes and the crossing over do not happen in mitosis. You would not want brand new different kinds of skin tomorrow that you have today. That'd be bad. But you do want a different child someday. You want them to be a little different than you. Unless you're Mr. Mason, then you want to clone them, right? I tell you guys I want a clone of me. Did I ever tell you guys that? It's my bucket list. I want to have a clone. Yeah. Well, I just think that like when I retire, someone should be here to take my job over, and he could do it, right? Be like, can you imagine bringing your kids back to school and then there's me again? But it wouldn't be me, but it would be. Yeah. That'd freak you out, right? Sit down. So therefore, a main difference between meiosis and mitosis is that meiosis has genetic information that is rearranged, leading to the variation in offspring. So the fact that you guys do that crossing over, and everybody does it, and you make new sperm and new eggs that are completely different than even half of you, gives us this whole new you when reproduction occurs. So this genetic variation is advantageous for a population. It gives us variety. It gives us new combinations of who we are. And this can help a population survive a major environmental change. And the only way this is possible is through sexual reproduction, mixing and recombining new gene combinations, making new versions of us. This is also what drives evolution. Which combinations survive better than others, right? I am sure that Inuits being colorblind, I always pick on Eskimos because they're in kind of a unique environment, but because they're slightly colorblind, probably had something to do with the lack of color up there. Oh, there's more colorblind people. So those genes weren't as important, right? So they didn't get chosen. But the ones that also made them have a little more fat helped them out too, okay? Whereas if you got maybe somebody that's, what's a hot region? Let's go to like Australia, right? Let's go there. Like Aborigines probably aren't going to make it too well up in the Arctic, right? Yeah. Okay. Different gene combinations were chosen for. So in sexual reproduction, existing genes are rearranged, and meiosis is the process that makes the rearranging of genes possible. Now the fusion of haploid cells from two different individuals adds further variation. And if I asked you, what are the two haploid cells called that fuse? What are they called? Gosh darn it. There. Oh. Click, please. And now you aren't even going to let me choose one. What does that call them? There we go. What are the haploid cells called? If you're haploid, you're 1N, right? What are the haploid cells called? Final answer in 5, 4, 3, 2, 1. No, germ cells are actually diploid. They're 2N. And diploid is kind of the opposite of haploid. Diploid is 2N. Sperm and egg are examples of haploid cells, but we call them gametes, right? And I'm trying to move down. I know some of you put gametes, but I'm having a problem with my board. Big problem. See, even my board is wore out. It's Friday, right? Here. See if anybody has it. No, a zygote's 2N. Zygote is nothing. Well, you know what? Oh, that's not. No, we won't get into that. Gametes, yes. Germ cells, no. Okay. What does that mean? Oh. So what? Clay? So during prophase 1, you guys told me the homologous chromosomes line up next to each other. Each of them is made of two sister chromatids attached at the centromere, and crossing over happens when one arm of one chromatid crosses over the other arm of the other chromatid. So if you have one chromosome here and you have another chromosome here, we understand that when they are next to each other, they actually swap bits and pieces of what they are. And then we have 1, 2, 3, 4 completely genetically different chromatids. Okay? This is, again, why all sperm are different than all other sperm, and all eggs are different than all other eggs. So the chromosomes then break at the point of the crossover, and each chromatid reforms its full length with a piece of the other chromosome. Thus, the sister chromatids of homologous chromosomes no longer will have genetically identical chromosomes. They'll be different, or information. So although they were identical when we started, once they pair up in homologs and do the crossing over, then each of those four is completely different. And we talked about independent assortment, right? Now during metaphase 1, homologous pairs of the chromosomes line up at the equator of the cell, and the two pairs of chromosomes can line up in either of the two equally probable ways. And a lot of you are like, I don't get that part, because it is a little confusing at first. I could line up like this, right? Or I could line up like this. I didn't say which way, did I? And that'll change. In all those 23 pairs in ours, each one could be lined up either or. So now imagine having 23 ways and the different combinations you can make of all of those. It's 23 to the, and there's two combinations. You'd have, oh boy, a lot of different combinations. That'd be what, 20, that'd be exponential, so it'd just be 23 squared, right? Yeah. So 23 times 23, whatever that is. That's how many different combinations there would be. And that's just that, without crossing over coming into play. Not to bring math into the situation. So this random distribution of homologous chromosomes during meiosis is what is called independent assortment, and we're going to deal a lot with that when we get into Mendelian genetics, okay? And that does require a minimal amount of math, just a little bit. And then, of course, the final randomness and the final biodiversity component is fertilization. This is a random process that adds genetic variation. The zygote that forms is made by a random joining of two gametes. And because fertilization of any egg by a sperm is random, the number of possible outcomes is squared. So I just want you to think, a female can carry up to like 400,000 eggs. One gets passed down a fallopian tube at a time. A male releases roughly 1 to 4 million sperm. One of those makes it. Just think about the randomness of that alone, okay? And then think of the fact that each of the sperm are different and each of the eggs are different. And now think about how different you actually are. That's immense. Now think about a salmon coming upstream, laying millions of eggs, and then the male salmon, which could be, 4 or 5 different male salmon, all releasing their sperm, which is called their spawn, okay, on top of it, and basically millions upon millions. And now you see the randomness that happens not only in us, but also in other organisms. So we talked about meiosis. This is where a diploid germ cell goes through 2 divisions to form those 4 haploid cells, and you guys are going to finish your drawings here in a minute. Mitosis produces cells that are used during growth, development, repair, and asexual reproduction, whereas meiosis makes cells that enable an organism to reproduce sexually, and it only happens in reproductive structures called gonads, testicles, and ovaries. The 3 contributions to genetic variation are crossing over, that happens in prophase 1, independent assortment, that happens in metaphase 1, and random fertilization, which happens during, well, depends on what type of organism you are. Okay? We know how humans do that, but then fish do it differently. So that fertilization component, even the birds and the bees, right, flowers in the trees, they do that differently as well with pollen. And okay. Do you guys know flowers have ovaries too? Yeah. Yep. All right. So I need you guys